Hello everyone, and welcome to this Genetic Engineering and Biotechnology News webinar. It's entitled Developing Relevant Epigenetic Models to Enhance Drug Discovery. In her wonderfully engrossing book, The Epigenetics Revolution, Dr. Nessa Carey calls the world of epigenetics a fascinating one, full of subtlety and complexity and influencing huge areas of biology. Indeed, Jen regularly reports on how scientists all over the globe continue to demonstrate that epigenetic modifications play a critical role in the incidence and progression of human disease. However, what's still needed are more precise and effective tools to study the epigenetic-based molecular events. Today's webinar will focus on a series of novel platform technologies from PerkinElmer that are used to measure epigenetic changes in model systems and to improve drug discovery research. John Sterling, Editor-in-Chief of GEN, and I'll be moderating the webinar. But first, let's meet our presenters. Dr. Michael Christopher Keogh works as Director of Markets and Application Development at Epi Titan. He's going to discuss the use of full-length histones and nucleosomes as more biologically relevant substrates for epigenetic enzymes. Roya Pedram-Fatemi is a doctoral candidate in the Molecular and Cellular Pharmacology program at the University of Miami Miller School of Medicine. Roya will describe her recent work on long, non-coding RNA epigenetic enzyme interactions as a novel class of drug targets for the upregulation of therapeutic genes. After the panelists make their presentations, there will be a question and answer segment. Feel free to send in a question for our panelists at any time during the webinar. Type your question into the Ask a Question box on the lower left of your console and then hit Submit. The panel will try to answer as many questions as possible. So if everyone's ready, let's get going. Dr. Michael Christopher Keogh will be our lead-off presenter. Michael?
Thank you, John. And just before I begin, I'd like to thank the organizers, obviously, at GEN, who've made this extremely easy. So as John mentioned, I represent Epicipher, and we're one of the components of we're an early stage in epigenetics company, so we're exclusively focused on the development of epigenetic reagents, both for academics and for industry. And one of the components of our work is, as he mentioned, with PerkinElmer, and I will present some of that work as a component of one of my slides. So today we're going to be talking about the development of relevant epigenetic models to enhance drug discovery, but also for academics. And that's essentially divided into looking at the creation and the rolling out of specific reagents that are required for various stages in sort of epigenetic investigation. So what is epigenetics? So I've been working in Okay, so we all know that the human genome isn't actually just packaged straight into the nucleus as naked DNA. It's actually associated with histone proteins, 8 core histones and a linker histone, into higher order structures known as chromatin, and then all the way up to the metaphase chromosome that we remember from our biology textbooks. This is a structure that's actually only seen at mitosis. So, but what is epigenetics? Well, epigenetics is actually it's the understanding that there's information encoded on top of the primary sequence. And this is information that seems to be held primarily within the proteins that actually compose the 8 histones and the linker histone. And it's because these actual proteins are modified. And these chemical modifications, which are essentially all reversible, act to distinguish specific regions of the genome. So this is a slide that we've been showing for years. This is one that was actually from a review back in 2002. And even then it was actually recognized that there were multiple modifications that were associated with the histone proteins. And the big ones that we are all familiar with are the acetylation on lysine, the methylation on lysine, either mono, di, or tri. Ubiquitination on lysine, methylation on arginine, either mono, asymmetric di, or symmetric di, and phosphorylation on serine. And these modifications are still probably the ones that are most understood by people and most well-studied. The readers and writers for them are pretty well understood by now. But what has been happening in the background in the last 10 years is actually an understanding or a discovery that there are multiple additional modifications, all the way from ribosylations, butyrylations, glutamylations. At this stage, probably the majority of these are still at the discovery stage, where they've been identified as post-translational modifications by mass spec analysis In some cases, the readers and writers are actually understood, but to be fair, the majority of the work, particularly in the drug development field, is still largely focused on the group on the left, the ones that have been actually known about for at least 10, 15 years. So what we know is that presumably these modifications, and this has been sort of guesstimated because obviously if they're being added and taken away and they're actually highlighting specific regions of the genome, they must be for something. So what we know is that there are enzymes that add them. These are known as the writers. There are proteins that distinguish and recognize and distinguish the modifications. These are known as the readers. And then there are erasers. From the perspective of the academics, it's obviously, it's extremely interesting to understand how these things are added, why they're added, when they're added, what are they for. For the purposes of drug discovery, with cancer genome atlas, etc., and obviously directed studies, it's been recognized that mutations in the writers, readers, and erasers are also associated with human disease. And this obviously gives an input because if you understand that a mutation is not simply a passenger but is actually a driver of human disease, you have an opportunity to actually target these things with inhibitors or activators depending on the nature of the mutation in a bid to actually alleviate the disorder. So if we look over the last probably 10 to 12 years, there has been a directed, concerted effort, now that the academic environment has actually identified the readers, writers, and erasers, to actually develop inhibitors against them to see if you can actually treat human disorders. So here we've but the thing is, if you actually look at the current field to date, we've identified in EpiCypher about 126 molecules that have been released with epigenetic targets. This is both drugs that are actually in human clinical testing and proof of principle molecules that are used to actually investigate the function of specific proteins. So these molecules aren't randomly distributed across the blocks of writers in red, readers in blue, and erasers in green. They're actually specifically focused or very heavily focused in specific subclasses in here. So what you can actually see is the bromodomain, for example, in blue gets 37. There's a significant drive to actually identify bromodomain inhibitors. And the protein methyltransferase class, which includes both arginine and lysine methyltransferases, there's also a significant drive there because a lot of these methyltransferases have again been associated quite tightly in a lot of cases with human disease. And in some cases, patients have actually been treated with these agents and have already shown promising responses. So if I just look at two of the classes with you, the first one is the bromodomain class, which contains 37. This class contains 37 molecules. 11 of these are in clinical trials. And what you see is if the bromodomains are divided up into usually 8 classes based on their sort of subsidiary structure. But if you notice, the 37 molecules that are in development are not randomly distributed through this. They're all heavily focused. What you can see is that of the class 2 is the one with the tightest focus. And this contains 25 of the molecules and all 11 that are currently in clinical trials. And the poster child for this group is BRD4. BRD4 work from years ago from Jay Bradner's group showed that there was an inhibitor called JQ1, which actually mimicked acetyllysine, and a bromodomain recognizes an acetyllysine. So this actually mimics acetyllysine and binds in the BRD4 bromodomain pockets and prevents its recognition. And it turns out that BRD4 turns MYC into a druggable target in some diseases because it's upstream of MYC as a transcriptional activator. So if you treat some MYC-dependent cancers with BRD4 inhibitors, you actually gain control over and actually have the potential of treating them. And some patients have shown responsiveness to this. If we have the next class, the arginine the lysine or protein methyltransferase class, this forms into two groups. The lysine methyltransferase is on the right and the arginine on the left. Again, there's 35 molecules that have shown promise as inhibitors within this group. And these are the arginine methyltransferase inhibitors, which I've highlighted here, and the lysine methyltransferases, which I've highlighted. There are 4 of these which are currently in clinical testing. The one on the left is DOT1, which is a lysine methyltransferase, but because of structure, actually looks more like arginine methyltransferases. So we've got a DOT1 inhibitor on the left. And then on the right, we have 3 EZH2 inhibitors. And EZH2 is a member of the polycomb repressive complex, which is responsible for H3K27 methylation. And Roya will talk about this in a few moments. or actually the next component, because she's going to discuss long non-coding RNAs that are actually inhibitors of EZH2 function, EZH2 targeting function. So if we move on, so in terms of epigenetic agents, how did we get to the sort of stage we're currently at? We usually look, and a lot of people actually look at this, is that there are probably at least 2 generations in terms of drugs that are epigenetic regulators. The first generation are the ones that currently contain approved drugs. These are agents which are currently being taken by patients, they're marketed, and they include the DNA methyltransferase inhibitors, which I haven't really spoken about DNA methylation, we won't go into it now, and then broad-spectrum lysine deacetylase inhibitors such as vorinostat. And the reason these are first generation is because, to be fair, any role they have in epigenetic regulation was actually discovered after the fact. The molecule was already well under study as a potential regulator of cellular function, and it was only later that was fully understood that it was actually an epigenetic regulator. The second generation are the current agents, and these fall into the BET inhibitors, which I mentioned, one of those that inhibit BRD4 function, and also the lysine and arginine methyltransferase inhibitors. And these agents were identified, it's a directed discovery approach, whereas you know that a specific molecule, or you know that a specific target is under study, you care about EZH2, you care about BRD4, and you're developing inhibitors in a targeted fashion directly towards those. The next generation is one that is currently probably only now beginning to be available. And these are substrate-dependent targets. So the one thing that will be noted is, I mentioned that the chromatin is essentially a nucleosomal-based structure. The idea is that you've got these higher-order structures of nucleosomes with sticking-out tails. And nucleosomes are not the same as peptides. And a lot of the early studies which were done on the development of the second-generation agents, actually these studies were done using peptide substrates. The opening up of new substrates, such as nucleosomes, will actually give you access to agents which are absolutely dependent on nucleosomes. They require their physiological template to bind or to actually carry out an enzymatic reaction. They will not do so in the presence of simple peptide substrates. And those are ones that we're currently working on within EpiCypher and in other companies to develop nucleosomal substrates for the actual study and development of these agents. So I've mentioned, I've sort of alluded to peptides versus nucleosomal substrates. So let's actually look at the 3 sort of potential groups that you could be working with. First, you've got histone peptides, which are obviously, as you'd imagine, 18 to 20 or 24 amino acids long. They're synthesized chemically. They're highly characterized because you know you could simply sticking residues together to make peptides. This technology has been around forever. They're easy to synthesize. They're easy to employ. You actually can add a bioassimilator or any immobilization agent and immobilize it onto beads. The only issue is, with regards to chromatin studies, they are low complexity. They cannot recapitulate a nucleosome. In some cases, of course, that might not be necessary, but for the new generations of agents, it probably is. You can sort of increase complexity in trans by making arrays of peptides. This allows you to sort of study multiple tails simultaneously, but again, it is simply just a collection of peptides. So this is used as a discovery tool both to characterize reader function and in some cases to characterize writer function as well, because it actually asks, if my enzyme is targeted to a specific modification, will it still work if an adjacent modification a priori exists? Or, and that's both with regards to readers, writers, and also with regards to post-translational modification-specific antibodies, and I'll give you 2 examples of that later. However, what we're always trying to get to are nucleosomes, because these are the physiological substrate. You can purify them. What you end up with, essentially, you break open cells and you purify the nucleons. What you end up with is a high-complexity substrate or template for downstream studies. But of course, it's non-defined because it contains the entire plethora of modifications that are present inside the cell at that time. So what you actually have is a population of post-translational modifications and relatively little control over that. You can skew it in certain directions by pre-treating the cell with various inhibitors. The classical one is HDAC inhibitors to drive up acetylation levels. But generally, it's not an optimal substrate for discovery. It also has non-trivial isolation protocols, and one of the concerns in downstream is because you're actually purifying the nucleosomes from cells, there's always the possibility of downstream contamination with the readers or writers or eraser enzymes that you want to do in downstream studies. And one of the classical science here is a lot of the early purifications of nucleosomes were identified actually to be contaminated with methyltransferases, which was obviously a major concern. To get around this, you actually want a blank slate. So what you would rather have are recombinant nucleosomes, which are defined, synthesized recombinantly, obviously, but with no post-translation modification. They're a blank slate, and these are very, very useful. And normally, they also allow you to do things like immobilize directly, because you can add a biotin group during the synthesis process or their purification. And the next generation of these, which we in EpiCypher are actually working with, is the creation of designer nucleosomes. And these are synthesized with site-specific post-translational modifications. You don't use an enzyme to add the modification to the recombinant. You actually synthesize a nucleosome which contains a defined modification. This obviously becomes an extremely useful tool, because you end up with 100% population, all of which contains a defined mod, which you can then use for binding, eraser, or reader studies. These are obviously relatively high complexity reagents, and they recapitulate the physiological substrate. So if we go back over the end, we can say, well, we can actually do a quick overview of these three reagent classes for the last part of my talk. I'll talk quickly about histone peptides, which we make available in either single or combinatorial site-specific modifications. The sample application for this will be the protein-protein interaction assays on the Alpha platform, and then I will talk about Epi Titan histone peptide arrays, and also then for the latter part of my talk about nucleosomal substrates, which we're going to use for the next generation of epigenetic targets. And the poster child for this generally is NSD2 methyltransferase. So to begin with, the first group is obviously the peptide substrates. So these are simple but highly characterized, and we currently have a library of around 250 histone peptides, which contain all of the current modifications, and this is always open to upgrading. So we're going back at all times and adding in new modifications like citrulation, butyrylation, etc. The peptides are available in single or combinatorial modifications, generally up to 6 per peptide. They're all HPLC purified and MS analyzed, and these are used for interaction or enzymatic studies. So all of the peptides are biotin-related, so you can actually use them for pull-downs and for enzymatic reactions. And one of the standard protein-protein interaction tools that are available in the to researchers is the AlphaAssay platform, which is available from PerkinElmer. And this, excuse me, this is a proximity-based assay which allows you to determine whether an interaction is taking place. So you can actually add whether the interaction is occurring a priori or to develop inhibitors to actually break this interaction. So it's a simple proximity-based assay which is analogous to FRET. It's sort of a chemical FRET. So you have a laser beam. So what you're essentially asking is the donor bead and the acceptor bead brought into proximity? And you can actually ask this by laser exciting the donor bead, which under this laser excitation at 680 nanometers releases single oxygen. If the acceptor bead is within a given distance from the donor bead at the time the excitation took place, single oxygen will diffuse across, bind to the singlet bead, and cause the emission of a light at 520 to 620. So it's a simple proximity-based assay. So here we're simply looking at to bridge the distance between the donor and the acceptor. We're actually using a biotin-related peptide which contains an acetylysine. And to this, our actual bridge is a his-tag bromodomain. So in other words, and the his-tag bromodomain is bound by the nickel chelate acceptor bead. So it's a very simple assay. So you can, once you actually see that the interaction is taking place, you can then add in inhibitors and ask whether they break the interaction between the bromodomain and the acetylated peptide. And here we're looking at one of the bromodomains of BRD4, bromodomain 1, and this node is known to bind to acetylated histone H4 peptide, either in a hyperacetylated form, the H4 tetraacetyl on the top, or monoacetylated on the bottom, H4K12 acetyl. And the inhibitor which I mentioned earlier, one of the earliest inhibitors that was identified of BRD4, which was characterized initially by Jay Bradner. And what you see is that as you titrate in JQ1 levels, the interaction between BRD4 bromodomain 1, and the tetracetyl peptide is broken, so therefore you can actually read this as the loss of the actual emission on the acceptor bead. So this is work that was done by Dan Cardillo in PerkinElmer. And in collaboration with PerkinElmer, we are actually releasing currently various AlphaAssay kits to initially identify the interaction between BRD4 and various acetylated H4 peptides, also P300, which also has a strong disease association. And over the next year, we'll be releasing a slew of kits to actually look at various epigenetic readers. And this is a potentially useful reagent to a great number of people, both in academic labs and also in the commercial environment. So if we move on to the second reagent class are the histone peptide arrays. And these have multiple uses, including validating PTM-specific antibodies and investigating the specificity of enzyme and reader domains. The current incarnation of our arrays contain around 250 peptides with single or multiple PTMs. And that's all that are currently available as individual peptides, and new printings of the arrays generally include novel modifications as they come online. Each peptide is represented 12 times per array in blocks of triplicates, and arrays are assembled by spotting biotinylated peptides onto a streptavidin-encoded slide, along with a tracer and extensive additional controls for the most likely means of downstream analysis. So in a standard analysis, a PTM-specific antibody or reader domain is added as a first layer, and this is the panel on the right. This binds to its preferred target, and that would be the appropriate array peptides containing the modification of interest. And areas of enrichment are then identified by an appropriate second layer fluorescent antibody. As mentioned, the primary use of these arrays is to examine the specificity of protein-protein interactions, and that's either by reader domain or PTM-specific antibodies. In the first example application, as epigenetic researchers, we're absolutely reliant on antibodies that can reliably distinguish and bind with high affinity specific PTMs. And obviously the keyword there is reliably. And some months ago, Scott Rothbard and Brian Strahl published an analysis of about 100 PTM-specific antibodies from various commercial sources. And this was published in Molecular Cell, with the entire data set also being fully searchable at histoneantibodies.com. So to give an example, you click on the histone you care about, and then the specific residue. And in this case, we're looking at a panel of 5 antibodies which are marketed as specific to H3K4 trimethyl. So the peptides containing this specific modification are circled in red, with yellow indicating strong binding on the array. And you can see huge variability in the specificity profile of each antibody. And this is obviously something to keep in mind when using each of these specific reagents. But a more common use of peptide arrays is to examine the specificity of enzyme or reader domains. So to give an example of that application, we'll use the arrays to do a survey of H3K9 methyl binding protein. So H3K9 is available in 3 methylated forms, mono, di, or tri, where H3K9 trimethyl is one of the canonical marks of heterochromatin, and multiple regulators are thought to be directly recruited to this PTM through various reader domains. And this includes UHRF1 via its tandem tutor, HP1 in various forms via its chromodomain, and MPP8 also via chromodomain. In contrast, H3K9 monomethyl is enriched at euchromatin, and this is reported to recruit GLP1, which is a methyltransferase, via its ankyrin repeats. And here we have array data from Brian Strahl's lab with the identity of each specific peptide in the horizontal and the binding domains I've just mentioned in the vertical. And going left to right, you've got the heterochromatin factors UHRF1, 3 paralogs of HP1 and MPP1, and on the far right, the euchromatin factor GLP. So brighter yellow is more intense binding, and what you can immediately see is the selectivity of the heterochromatin factors for peptides that contain H3K9 trimethyl, where GLP doesn't bind here, but instead it binds to a peptide containing H3K9 monomethyl. But what you can also see is that most of the heterochromatin factors, such as HP1, for example, can bind to H3K9 trimethyl peptides that also contain H3 serine 10 phosphorylation. And this supports previous findings where the phosphorylation of H3 serine 10 by aurora kinase, which occurs at mitosis, displaces multiple heterochromatin factors and acts as a phospho switch to control the reformatting of heterochromatin through to the next cell cycle. The final reagent class I'm going to talk about are nucleosomes, and these are higher complexity substrates which are more representative of the physiological state and give you access to families of enzymes that have thus far proven resistant to investigation because they're unable to target peptides. And one example of this is the NSD2 methyltransferase. Nucleosomes are generally available in two forms, either purified, examples being from the chicken or HeLa cells, which contain all the modifications that are pre-existing in the cell at the time of lysis, or recombinants, which represent a blank slate with no PTMs. And in our case, we generally provide the latter as mononucleosomes on the 601 positioning sequence, plus or minus a biotin tag for immobilization. So the reason for actually wanting access to these rather than continuously, exclusively focusing on peptides is, just to give you a quick example, many methyltransferases, lysine methyltransferases, actually display a strong preference for a nucleosomal substrate. I've just given you three examples. G9A, which is obviously nucleosome, it doesn't care, it'll free a nucleosome, or sorry, it'll methylate both free H3 and a nucleosome. NSD2 obviously shows a strong preference for a nucleosome over free H3, and SMYD2 shows the contrary. Now this could obviously give you an indication of the physiological role of the enzyme. Obviously SMYD2, its inability to target a nucleosome obviously tells you a great deal about its potential role inside the cell. Maybe it's a predeposition modification, or maybe it's actually targeting its preferred substrate is actually not a nucleosome at all, or even a histone. It might actually be a non-histone substrate. So just give you some of the QC data that we generally do. Here's just another example of this. This is NSD2 in giving it a nucleosomal or an octamer substrate. You see it shows a strong preference for the nucleosome of the octamer. We see exactly the same preference for set 8, which prefers a nucleosome over an octamer. And finally, the same preference for dot 1, which again prefers a nucleosome over an octamer. There is some argument in the literature that dot 1 will actually, methylate octamers, free octamers, but it turns out that this is actually an allosteric effect, which is induced by the presence of free DNA in the same reaction. So the preferred physiological substrate for dot 1 is actually also a nucleosome. So one of the proteins or epigenetic writers that was identified years ago as associated, very strongly associated with disease, was NSD2, nuclear set domain containing protein 2, which is also known as MMSET, multiple myeloma associated set protein. And this is because there's a recurrent translocation that occurs, which moves NSD2, it's the 414 translocation, which removes NSD2 from its usual location and puts it downstream of the strong c-mu gene promoter. And what this does, the translocation is observed in multiple myeloma in about 15 to 20% of patients, which fall into the worst prognostic group. It's not a break fusion. It's actually an overexpression. And it's now known that NSD2 is overexpressed in multiple cancers, and also that an activating mutation, E1099K, which is seen in pediatric ALL. So actually, NSD2 is known to act as a general oncoprotein in transformation assays, and this requires its catalytic activity. So for years, people were trying to actually study it in terms of this, and one of the issues that hampered all the discovery-based approaches was that the actual catalytic activity of NSD2 wasn't fully grasped or fully understood, and this was because the early attempts had all tried to use peptide substrates, which suggested that it had an activity towards multiple targets, including HTK4, HTK4, K27, K20, etc. But its actual physiological activity is HTK36, and this was identified when you actually look at nucleosomes. So work from Orgozani's lab is actually given nucleosomes to NSD2, methylated them in vitro, and then use mass spec or Western blotting to actually identify the preferred methylation events, and they are exclusively mono and dimethyl of H3K36. What you actually understood that nucleosomes are required to do this sort of work, you actually understand exactly what complexity is going to be required for you to do drug discovery. But work that we and others have collaborators in Minkyu Lui's lab Once we knew that NSD2 was a nucleosomal-dependent enzyme, you can actually start to create assays out of this and do the early stages that would be required to start to do inhibitor development. Here we've just added a nucleosome, immobilized it, and we're doing a radiolabeling assay with hot S-adenosine methionine to actually dial in on the optimal enzyme activity or the optimal domain or the reduced domain that are required to actually build an assay. Here we're looking at the NSD2 catalytic domain and showing that activity is a function of enzyme concentration. It's also a function of time. Because if you have control of the assay to that degree, you can actually start to do inhibitor development. So here we're simply doing a small proof of principle screen with an analog panel for sinfungin, which is the general methyltransferase inhibitor. So you see that in these circles, that sinfungin is a pretty poor inhibitor of NSD2, but a simple size substitution of a propanol group gives you selectivity. You actually get inhibition in the micromolar range of NSD2 with no inhibition towards .1, .7, and .8. So this is obviously a micromolar inhibitor, which is not a great deal of use as a drug target, but obviously it shows you that it is potentially possible now to actually develop inhibitors against this target. A broader screen is now ongoing between us and Q Lee's lab. In addition to, you can actually go back and rather than the radiolabel, you can actually start to develop an alpha screen. And this is, as I mentioned earlier, on the alpha platform from PerkinElmer. And here we're again looking at the readout of mono and dimethyl K36. Again, immobilized. So work with Orgozani's lab in Stanford has developed an alpha screen in a 384-well 16 microliter reaction volume, which can be further miniaturized to a 1536. So we're now at the high throughput stage. Here the enzyme is recombinant NSD2, the catalytic domain, which is highly active, and the substrate is recombinant nucleosomes. Now, there are future studies that we and others will probably want to do to incorporate designer nucleosomes, and this is because in vivo, the optimal substrate for NSD2 is probably not a non-methylated lysine at K36. It's probably a monomethylated lysine at K36, and this is sort of work that I don't really have time to go into at the moment. But this sort of observation is one of the driving forces behind our release of designer and variant nucleosomes, which contain either site-specific PTMs or histone variants. So within these classes, each specific D-nuc and V-nuc reagent is fully synthetic and therefore defined and homogeneous, and each of them is directly related to the recombinant mononucleosomes that we assemble on the 601 positioning sequence. So the table lists our current designer nucleosome release schedule to early 2016 and includes potential and obviously more physiological acetylysine substrates for the BEF-bromo domains and also methyl substrates for methyl binding domains and demethylases. And on the bottom is the V-nuc collection, and I just mentioned these a second ago. This contains a pretty broad collection of histone variants. And we've not really had time to go into these today, but the variants are specialized non-allelic replacement histones that often have unique deposition machineries and important roles in chromatin function, and now also have been a lot of them have also been implicated in cancer progression with the cancer genome atlas, etc. So as a recent example, the most recent example is with H2AZ.2, which has been identified as a driver of malignant melanoma. So we're currently or continuously adding to this pipeline, and over the next year or so, we'll also begin to release additional types of modification, such as arginine methylation, along with combinatorial designer nukes, which contain post-translational modifications on more than one histone. To end, I'd like to thank all of the collaborators who generated much of the data I've presented today, and this includes the Bedford, Gozani, Wu, and Strahl labs, along with Dan Cardello at PerkinElmer. And thank you all for your attention, and now back to John and Roya.
Thank you, Michael, for that very detailed account of how to develop and apply relevant epigenetic models to improve and actually enhance drug discovery efforts. Drug discovery is a cornerstone of new product development, so thanks so much for sharing your drug discovery insights with our audience. If you're just now joining our webinar, thanks for tuning in. Just a reminder that there will be a question and answer segment after the last presentation. Please put your question in the box on the lower left hand of your console and then hit submit. Our next speaker is Roya Pejam-Fatemi. Roya, you are set to go.
Hello, everybody. Thank you, John, and thank you to the organizers for including our work in this informative webinar. I'll be speaking today about our work on the detection of long non-coding RNA protein interactions using AlphaScreen technology. And just to summarize my talk, first I'll be describing long non-coding RNAs and why they are functionally important. And then I'm going to describe one of the mechanisms by which they function, and that is with epigenetic enzymes to alter gene expression. And finally, I'll be describing some of our work on assaying long non-coding RNA protein interactions for the purpose of drug discovery. Now, so what are these long non-coding RNAs, and why do we actually study them? Well, it seems as though the eukaryotic transcriptome is much more complex than previously thought. Many large-scale transcriptomic efforts have really shown this in the past decade or so. And this figure really sums up why we focus on the study of long non-coding RNAs. As you look at genome size, as genome size increases, we see the red boxes indicating that there's a general level off of protein coding genes. However, if you look at non-coding RNAs, non-coding RNAs seem to scale with genome complexity. And really, this is an unexpected deviation from the central dogma. We find that many of these non-coding RNAs are actually functional, and we look at non-coding RNAs more so as functional units within the cell as opposed to the faithful intermediary between DNA and protein as they were classically classified as. We also learn that genomic loci are much more complex than we previously thought, and we see that there are many novel non-coding RNA species, which I'll describe to you shortly. And we also see, interestingly, that many both protein coding and non-protein coding RNAs can arise from the same locus. And again, recent evidence has shown that many of these non-coding RNAs are actually functional. However, we're really only just beginning to truly understand and appreciate the functional role of these non-coding RNAs in the genome. So non-protein coding RNAs are actually divided into two basic classes. There's the small non-coding RNAs, which are less than 200 nucleotides in length, and these include well-known examples such as microRNAs, siRNAs, Pee-wee interacting RNAs, and small nuclear RNAs. And long non-coding RNAs, which are greater than 200 nucleotides in length, and these include examples such as natural antisense transcripts and long intergenic non-coding RNAs. And the focus of my talk today will be on a natural antisense transcript. And briefly, natural antisense transcripts are transcripts that arise from the opposite strand as that of a protein coding gene. And these transcripts, interestingly, can possess a 5' cap. They can be polyadenylated, and they can be spliced similar to messenger RNAs. And furthermore, they can be localized either to the nucleus or the cytosol. One of the ways by which long non-coding RNAs function is by orchestrating key epigenetic events within the cell. And there are two main interactions that enable long non-coding RNAs to guide epigenetic enzymes to specific genomic loci. First, a long non-coding RNA, such as a natural antisense transcript, can interact with an epigenetic enzyme, and in doing so, then interacts with a specific region of chromatin, enabling the chromatin-modifying enzyme to come into contact with neighboring histone tails. And depending on which chromatin-modifying enzyme it is and which mark is deposited on the histone tail, it will result in either gene repression or gene activation. And specifically, our work focuses on the non-coding RNA protein interaction highlighted here, and I'll discuss more about this in detail. Specifically, we're interested in the polycomb repressive complex 2, or PRC2, which Michael introduced earlier. This protein catalyzes the trimethylation of histone 3 lysine 27 to suppress gene expression, and it's made up of 4 core proteins. That's the RBAP48, SUS12, EZH2, and EED proteins. And our work focuses on EZH2. EZH2 is a histone methyltransferase that contains the catalytic set domain. And the structure of EZH2 is fairly disorganized. It's a 746 amino acid long protein, and the C-terminus, and in the C-terminus resides the set domain. And interestingly, there's also in red a non-coding RNA binding domain, which was discovered several years back. And this is where the non-coding RNAs are thought to interact with EZH2. And I'd like to point out that this RNA binding domain is actually separate and distinct from the catalytic set domain. So long non-coding RNAs can recruit PRC2 to regulate chromatin shape and gene expression, and I'm going to discuss a number of examples of these. But briefly again, PRC2 can interact with a long non-coding RNA and be guided to specific genomic loci where it results in a change in chromatin state from an open chromatin state to a closed repressive chromatin state. And a number of examples of this have been shown in the literature. For example, in X chromosome inactivation with the long non-coding RNA exist. In the case of imprinting with KCNQ1-OT1. In the control of the tumor suppressor INK4B R4 INK4A locus with ANRO, as well as the control of homeoboxy cluster genes with the long non-coding RNA hot air, which I'll describe later on in the talk, as well as in the control of the BDNF locus through the non-coding RNA BDNF antisense. Our work focuses on brain-derived neurotrophic factor, or BDNF, and this is a secreted nerve growth factor, and this protein is required for early neural development as well as maintenance of the nervous system. And BDNF is actually found to be downregulated in a number of neuropsychiatric and neurodegenerative disorders, for example, clinical depression, Alzheimer's disease, Parkinson's disease, Rett syndrome, etc. And our group interestingly found that the BDNF locus is actually epigenetically controlled by the long non-coding antisense RNA, BDNF antisense. The antisense transcript of BDNF, or BDNF antisense, is a multi-exonic transcript that's conserved in humans and in mice. And in the figure here, you can see that it's transcribed from the opposite strand as that of the BDNF mRNA. Our group studied BDNF antisense, and we observed that a knockdown of BDNF antisense actually results in a significant increase in BDNF mRNA, and this actually translates in vivo at the protein level. And so we were interested then in understanding the mechanism by which this repression occurs. And so we performed chromatin immunoprecipitation experiments on BDNF antisense depleted samples, and we observed that there's a reduction in H3K27 trimethylation specifically at the BDNF promoter, and this correlated with a reduction in chromatin occupancy specifically again at the BDNF promoter. And this really prompted us to study the mechanism and look at this as an immediate drug target. And this is our model of antisense-mediated epigenetic regulation at the BDNF locus. When BDNF antisense and EZH2 interact, it enables EZH2 to repress the BDNF locus. And by disturbing this interaction, we see a de-repression and a consequent upregulation in BDNF, again, both at the mRNA and at the protein level. EZH2 is an important drug target. It's overexpressed in a number of cancers, for example, breast and prostate cancers. However, the major drawback to studying or to targeting BDNF, to targeting EZH2 rather, has been that it's difficult to target this protein because of its loose structure aside from the catalytic set domain. And furthermore, efforts to target the catalytic domain of EZH2 can have a number of side effects, especially when you're targeting this ubiquitously expressed epigenetic enzyme in the brain. There have also been a number of RNA targeting strategies that have been used in the past. Oligonucleotide-based approaches such as siRNAs and modified antisense oligonucleotides or antagonists have been used with success in vitro, and some of these have actually translated over to clinical trials, late-stage clinical trials. However, one of the drawbacks of this approach is that delivery remains an issue. There's also the use of lenti, adeno, or retroviral approaches to deposit shRNAs to ectopically express BDNF. However, using viral therapies has a number of drawbacks, and it remains to be seen whether they can actually be used successfully in humans. And recently, the use of the CRISPR-Cas9 system has received a lot of attention. This is an RNA-based endonuclease system whose effects really remain to be seen, and whether or not it can be used therapeutically in humans really remains to be seen. And finally, there's the use of small molecules to target RNAs. However, small molecules targeting RNAs have really not been successfully observed. The best example of this is the use of aminoglycosides to target the 16S ribosomal RNA. And furthermore, attempts to deliver recombinant BDNF have also failed. So BDNF levels are tightly regulated within neural circuits, and overexpression of BDNF non-specifically can result in excitotoxicity as well as tumors and cardiovascular side effects. And recombinant BDNF level delivery methods, for example, intraperitoneal, intrathecal, and intranasal administration, have also shown to be ineffective even in later stage clinical trials, mainly because delivery and dosage seem to be a problem. And people have tried recently the use of BDNF mimetics, for example, TrkB agonists. However, the effectiveness of these therapies really remains to be seen. So our approach is quite different. So I mentioned to you the difficulties in targeting either a protein, specifically EZH2, or RNA. And what we think is that our approach has two levels that make it superior to targeting either a protein or an RNA alone. First, non-coding RNAs are inherently cell, tissue, and development time point specific. As such, we think that the BDNF antisense is expressed within certain cells, and so targeting this RNA will enable us to achieve one level of specificity. Second, our approach is to target the non-coding RNA binding domain of EZH2 rather than the catalytic set domain. So in targeting this non-coding RNA protein interaction specifically, as opposed to targeting the protein or the RNA alone, we think will provide a higher level of specificity and also have less side effects than targeting either component alone. However, in order to target RNA-protein interactions, we first needed a method to be able to assay and quantify these long non-coding RNA-protein interactions to screen for small molecule modulators. There already exist a number of methods to test for RNA-protein interactions, and these include things such as RNA pull-down under native or cross-linking conditions, followed by sequencing to determine which RNAs are interacting with specific proteins. There's RNA-EMSA, which allows you to see a shift in migration in an RNA through a gel when it's complexed with a protein. There's also fluorescence in situ hybridization, followed by immunofluorescence, which shows you which is a microscopic technique. It allows you to see a co-localization with the RNA and a protein. And finally, there's an RNase protection or a footprinting assay where you can essentially incubate an RNA-protein complex with RNases and observe the regions of the RNA that are masked by the presence of a protein. However, the major limitation with all of these approaches is that they're quite cumbersome. There's a considerable amount of time and optimization that's required to study these RNA-protein interactions. And really, none of these methods are amenable to high-throughput screening. That said, we use AlphaScreen technology from PerkinElmer to develop a high-throughput method by which to quantify this class of long non-coding RNA protein interactions. And we decided to use a cell-free system. We have a purified C-terminal flag-tagged EZH2 and that we incubate with a 3' end biotinylated non-coding RNA. And we use AlphaScreen to quantify this interaction. And Michael went through and discussed the AlphaScreen assay, so I won't repeat that. So we had to optimize our AlphaScreen assay. We tested we cross-titrated several RNA and protein concentrations, and we see that an increase in RNA results in an increase in AlphaScreen emission in a concentration-dependent manner. And I've circled in red here the assay conditions that we decided to use for later screening purposes. To ensure the specificity of our reaction, we had a negative RNA control, and this consisted of a vanilloluciferase transcript, and this transcript was not expected to bind to EZH2. However, we do see some nonspecific binding that we confirmed with RNA EMSA, and this is consistent with previous reports that EZH2 can bind non-mammalian RNAs with low affinity. Next, we tested a positive RNA control for EZH2. We tested the long non-coding RNA HOX antisense intergenic RNA, or HOT-AIR. This is a 2.1 kb non-coding RNA that interacts with EZH2 to repress several genomic loci. HOT-AIR, importantly, is a cancer target, and it's upregulated in breast, ovarian, and gastric cancers. And we see that HOT-AIR, similar to EZH2, increases the AlphaScreen emission signal in a RNA concentration-dependent manner. So next, we had a negative protein control for our assay, and this consisted of phosphofructokinase protein, or PFKP. This is a kinase that's involved in glycolysis, and it's similar both in size and isoelectric point to EZH2, and we didn't predict that this protein would bind to any of our long non-coding RNAs, and as expected, we see that both BDNF antisense and HOT-AIR are able to bind EZH2, whereas they're not able to bind to our protein control. Next, we tested our assay suitability for high-throughput screening. We looked at DMSO tolerance, and we found that our assay components, all AlphaScreen reagents, as well as our long non-coding RNAs, were stable in DMSO up to 1%, and we screened at a level that was tenfold below that. And we also looked at Z-factor, coefficient of variation, and EC50, and we noticed that over a several-day period, our Z-factors were consistently above 0.5, our variation was consistently below 10%, and our EC50 values were in the same range. So after performing those optimizations, we began high-throughput screening. We screened the Presswick phytochemical library. This is a 320 compound library that we screened at 10 micromolar, 0.1% DMSO. We used an EZH2 concentration of 4 nanomolar and RNA at 0.3 nanomolar. And we were very interested in using this phytochemical library specifically because of the recent emergence of natural compounds as therapeutics due to their increased drug-like nature. And also, in the past 30-some odd years, we observed that about a third of FDA-approved drugs were actually natural compounds or derivatives of natural compounds. And interestingly, just a couple of weeks ago, the Nobel Prize in Medicine was awarded to a natural compound that is an anti-malarial. So our primary screen consisted of the BDNF, of screening the phytochemical library against the BDNF antisense EZH2 interaction, and we used a cutoff of about 70% inhibition for our hits. And in red, we have circled biotin. Biotin served as an assay validation. Free biotin will bind the streptavidin-coated beads and prevent the association of BDNF antisense with EZH2. So our primary screen hits are listed in the table here. The compounds in blue, actually, we were not able to test due to limited compound availability, as well as the structures of these compounds. We've tested all of these hits, and we were able to validate 4 of the hits in a concentration with concentration response curves. And of the hits validated, only elliptocene was able to alter BDNF transcription in cells. And elliptocene is actually a plant extract. It's one of the simplest naturally occurring alkaloids. Mechanistically, it functions as a DNA topoisomerase II inhibitor, and derivatives of this compound actually possess anti-tumor and anti-HIV activity. And despite hydrophobicity and toxicity issues, derivatives of this compound are still being synthesized and studied. We tested this compound in cells, and we observed that there was a roughly 2.5-fold upregulation in BDNF mRNA when treating HEC-293 cells with 1 micromolar ellipticine for 48 hours, and this translated actually to a modest but a roughly 10% increase in BDNF protein. We also tested ellipticine in a more relevant target, in a more relevant cell model. We have a human neural cell model, a cell culture model in our lab, and we were able to see a roughly 6-fold increase in BDNF mRNA. Unfortunately, we were not able to test protein due to the limited amount of protein on the cells. However, we are still following up on this compound. We also performed a primary screen for hot air EZH2. We used a 70% inhibition as our hit cutoff. And again, we see the red circle indicates biotin, which was an assay validation showing that free biotin was able to inhibit assay signal. So we selected compound hits that were, we removed compounds that were hits for both screens, and we tested several compounds that were unique to hot air, and the compounds as well as their structures are listed. We were able to obtain and test all of these compounds. And we were able to validate one compound, camptothecin, as a hit for hot air EZH2, and we tested this compound in vitro. Camptothecin is a tree-derived quinoline alkaloid. Mechanistically, it functions as a DNA topoisomerase 1 inhibitor, and the most famous derivative of this compound is topotecan, which is an FDA-approved chemotherapeutic agent. Unfortunately, camptothecin itself is water insoluble and has many side effects, but the derivatives are still being pursued. And because of these negative qualities, in vitro studies in HeLa cells were not successful. Camptothecin was highly toxic and altered a number of housekeeping genes, and so we stopped working on this compound. So briefly to summarize my talk, we observed that non-coding RNAs such as BDNF antisense confer specificity to ubiquitously express epigenetic enzymes to regulate locus-specific gene expression. And using AlphaScreen technology, we were actually able to develop the first assay to measure non-coding RNA protein interactions for the discovery of small molecule modulators. And finally, our assay can be modified to measure specific interactions between multiple non-coding RNAs and their respective protein binding partners, which could have many future implications. And finally, I really want to express that non-coding RNA protein interactions represent an entirely new and unexplored field of druggable targets because of the reasons that I mentioned could have many applications in the future. Finally, I'd like to thank my dissertation advisors, Dr. Mohammed Fagihi, Dr. Klaus Wallstead, and Dr. Salahuddin, Dr. Brothers, and Dr. Volmar for their important contributions to this work. Our funding sources, the American Heart Association and the NIH. And finally, at PerkinElmer, Jimmy Barbie, Roger Bossé, and Evelyn Griffin for all of their help and time. And with that, I'll refer you back to John.
Thank you, Roya, and thanks again, Michael, for 2 excellent presentations on developing and actually working with epigenetic models to advance new drug discovery. It looks like you both certainly gained the attention of our audience based on the questions we received for our Q&A, so let's take the first one.
Okay, so our first question is for Michael. Michael, one of our audience members wants to know, how transferable are the standard high-throughput protein-protein interaction approaches? For example, AlphaAssay, TR-FRET, from peptide substrates to nucleosomes.
The quick answer is it should be pretty straightforward, and because the distances that are involved across the nucleosome from the reader to the output beads should have no problem diffusing over those distances.
All right, thank you very much. Michael, so our next question is for Roya. Roya, one audience member would like to know, could the compounds identified from your screen have effects on transcription at other gene loci?
That's a great question. Thank you for that. That is actually possible. We are in the process of testing this, but we don't rule that out by any means.
All righty, thank you, Roya. Michael, back to you. What are the plans for the histoneantibodies.com site? Will it be updated with new antibodies? Is all the data from you, or do you take external submissions?
So it has actually been updated since it went live so that it is now larger than it was when the molecular cell paper was published. And that includes data from both Brian and Scott's labs, and now there is a means by which you can upload your own data. It'll go through a QC phase to confirm that it's compatible with what they're doing. But the plan is, it's a fantastic resource. I mean, the guys have done a great job. It's a great resource to the community, so it will be a waste not to keep it going and to keep adding to it. So that's the intention.
All righty, thank you, Michael. Roya, another question for you. Do you know if your compounds can bind EZH2 or BDNF AS?
So that's another great question. Thank you for that. We are actually in the process of testing this as well. We believe it's a little bit tricky to test because we don't have any type of structural information for either BDNF antisense or EZH2's non-coding RNA binding domain yet. However, this is a point that we're working to address.
All right, thank you, Roya. Michael, in general, how do readers influence the activity of surrounding epigenetics enzyme, for example, erasers, writers, associated to the same cluster or scaffold?
It really depends. One of the things that you notice when you start working with epigenetic complexes is they're generally extremely large. I mean, these readers don't wander around as monomeric proteins. The proteins themselves generally sometimes contain multiple reader domains, but every reader complex will contain multiple domains, either on one reader element or diffused throughout. And the complexes generally also contain the ability to contain writer or eraser domains. So what you occasionally see, well, actually it's very common to see a thing called an amplification loop. In the example that I gave for GLP, which contains anchorin repeats which are specific for monomethylated K9. That is also a methyltransferase, which contains the activity towards H3K9 monomethyl. So by the very act of recruiting, it stabilizes itself by methylating around it. And that sort of example is seen throughout. So you'll also get occasions where it'll the reader domain will recruit the eraser, so that way it's a short-lived signal.
Okay, thank you, Michael. Roya, the next question for you. A reader wants to know, or audience member wants to know, excuse me, are long non-coding RNAs the main drivers for epigenetics machinery, specificity at the DNA level?
So interestingly, it's been shown that the enzymatic components of epigenetic complexes actually have RNA but not DNA binding domains. So that really strongly suggests that these RNAs play an important role in the recognition event. And furthermore, studies at the molecular level have shown that these enzymes can bind a number of long non-coding RNAs. For example, exist, hot air, KCNQ10T1, BDNF antisense as I presented, etc.
All right, thank you. Michael, question for you. Will the DNUC and VNUC be available in the AlphaAssay high throughput screen?
The answer to that is yes. There's a bit more work to put the screens together than for a peptide, but that is the general intent. I mean, when we put these together, we're going to try and make them compatible with direct research that researchers want to do, but also with high throughput screening. So we have it in mind that since they are compatible, so that would be the intention for all of them going forward.
All righty, thank you, Michael. Roya, we have another question for you. Why is there such a large difference between the effect of ellipticin on BDNF mRNA and protein expression levels?
So that's a really great point. We think that there may be an issue whereby the BDNF is being produced. However, BDNF is actually, it's a protein that's processed several times before it's actually, it actually leaves the cell. So we think that possibly there's some of the BDNF protein that's being processed. However, it's not able to exit the cell. And I've performed actually Western blots to try to determine if there's anything inside of the cell. However, those results have kind of been inconclusive so far. But in terms of what's actually secreted into the media and what's actually processed, it seems that there is some BDNF protein. It's a modest increase, but it's something which signals that that this mechanism is working.
All righty, thank you, Roya. Michael, we have another question for you. Our audience member wants to know, usually epigenetic assays involving tags such as antibodies, are reported to be a problem. Are the peptides and nucleosome preps compatible with HTS mass spec screening for drug discovery?
There's probably two answers to that. The first one is the original statement. He's exactly right that some of them can be a problem, and the reason for that's if you use a dodgy antibody. And you can see, I mean, if you look at the histoneantibodies.com, some of the antibodies that are out there that are ostensibly sold as specific for a given modification are either not as specific as you think, or they're heavily influenced by adjacent modifications. You get this combinatorial problem where it's raised against residue X, but it doesn't work if the neighboring residue is modified. So this is one of the reasons in sort of there's this apocryphal work or data going around, oh, this approach isn't that great. It really does depend on the reagents you start with. But to get to the second part is, are the peptides and nucleosomes compatible with high throughput mass spec? And the answer, of course, is yes. You're going to get a bit more carry through with nucleosomes because obviously you're going to get the peptides from that, but there's no reason not to make it compatible with those approaches.
Thank you, Michael. Roya, looks like we have another question for you. Can you use the system to screen for proteins interacting with a given non-coding RNA?
So absolutely, we think so. I've showed you the conditions that we've optimized for just a couple of proteins and RNAs, but there's no reason, I don't think, why you can't, with the proper tags, be able to use any purified protein or non-coding RNA to test for an interaction.
Righty, thank you, Roya, but don't go anywhere. Looks like our last question for the webinar is going to be for you. Our audience member asked, could compounds targeting the long non-coding RNA epigenetic enzymes interactions be more specific drug candidates in general? Example, when compared to writers or erasers inhibitors.
So we absolutely believe this. We think that targeting long non-coding RNAs specifically at their site of interaction with EZH2 can alter genomic loci without altering the other effects of this important enzyme in the cell. And the reason we believe this can be done is because EZH2 actually has a separate and distinct non-coding RNA binding domain that is functionally, it's far apart from the catalytic domain, and so we think that again, by targeting this binding domain, we're altering genomic loci without blocking or altering any of this enzyme's other important functions within the cell.
All right, thank you very much. And with that, it looks like we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived for 6 months on our website at www.genengnews.com. So if you missed parts of it, you can watch it again, or feel free to forward the link to your friends and colleagues, which we always recommend. I'd like to thank Michael and Roya again for their informative presentations, and I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to PerkinElmer for sponsoring this webinar. So hopefully we'll see you again in another Gen webinar in the near future. Goodbye for now.